Good afternoon, everybody. I'm Jayatri Das, Chief Bioscientist here at the Franklin Institute. And thanks for joining us for one of our monthly updates around what's going on in the constantly changing world of the coronavirus. It's my honor today to be joined by Dr. Guy Padbury, who's the Senior Vice President of Preclinical Development at Merck. And we're going to get into a little bit of what that means in terms of his work and talk about what insights he has into the process of actually making the vaccines at the scale that we need to protect people around the globe, because that's a lot of work. And it's a little mysterious about how that all happens. And as we're reading in the headlines today, getting that up to scale is more important than ever. So send us your questions around that. Before we get into it, though, I wanted to talk about some of the other aspects of the virus that has been really hitting the news in the last couple of weeks. Just before Christmas, we started to learn about some variants of the virus. Now, this is something that we've talked about on some of our conversations before, Guy, and maybe you can help us think through what's going on here, is that the virus seems to be changing. And there are a couple of versions of the virus now that seem to actually behave a little bit differently from the original version. So do you want to help us bring us up to speed in terms of what we're looking at with these new versions?
Right. Yeah, a very timely question, right? I think these are generally referred to in the press as either the UK or the English strain, or the South African strain. And I've lost track over the weekend, but we're something in 5 or 6 states here in the US have now identified that strain. The general understanding at this point in time is those two variant strains. Well, let me back up. Viruses mutate all the time. A lot of them are silent mutations, which have no impact on the virulence or the severity of the viral infection. And then others, like we're experiencing with the English and the South African variants, in this case, they don't seem to increase the severity of the infection, but their transmissibility seems to be higher across populations.
So what that means is that it doesn't, if you contract it, it doesn't make you any sicker. It's just easier to catch it.
It's just easier for me to give it to you, or vice versa, or our friends and family, which then increases the societal risk, right? Rather than the individual risk. At this point in time, I think we're still in the learning phase. It's very early days. What I understand, not being a virologist, but is that in particular, the key focus is on the amino acid identified as 501 in the spike protein, which is the cell surface protein on the virus, which the vaccines are targeted against. There is a mutation that changes that from an asparagine amino acid to a tyrosine. And that in news that came out from Pfizer on Friday, that hasn't been peer reviewed yet, but I think is credible, is they have constructed that specific mutation in the test tube, and using blood samples from 20 or so patients have shown that their vaccine is still active against that variant. Now, there may be other mutations associated with that, so further studies are ongoing, but I think that that is a good early read that even though this mutation has an impact on the transmissibility, it will still be susceptible to the vaccine. And in particular, obviously, there's the data on the Pfizer vaccine, which is the mRNA vaccine. But I think there's been a lot of concordance between what we're seeing with the Pfizer vaccine and the Moderna vaccine, which is also an mRNA vaccine.
All right, that was a great overview. So I want to dig in a little bit into a couple of the different points that you covered. So I think where you started, I think is something really important to think about, is that different variants come up all the time. There have been lots of different versions that have been tracked all over the world. And the way that scientists find that is by sequencing samples of the virus.
Right.
And one of the biggest efforts in tracking how the virus is changing has been going on in the UK. And that's one of the reasons that this was discovered so quickly. and now it's been popping up all around the world. So why this one? You mentioned this particular amino acid change. We've often talked about how viruses and the receptors in our cells are kind of like a lock and key mechanism where that spike protein of the virus is like the key that fits into the lock. So what people think is that this particular change in that amino acid sequence that you mentioned, it's almost like it's a better key that fits tighter in the lock. Is that a fair analogy?
Yeah, I think the structural reactivity effect yet of this mutation is yet to be understood, right? In some cases, it changes a conformation, which is kind of your key and lock analogy. I think in this case is the science is yet to be discovered of what is it about this mutation that changes its activity profile, that allows it to be more transmissible than others. And, I think unlike small molecule, drug discovery, where a lot of those key and lock and ligand receptor things are well understood, even though we've had vaccines for, many decades now, the understanding of vaccines at a molecular level is still relatively in its adolescence. I won't say infancy, I'll say it's adolescent, right? And again, as each one of these viruses that we get exposure to teaches us more about what questions we still don't know the answers to, right? So I think at this point in time, at least what I've seen largely in the public literature is that it's not exactly clear what this mutation does to elicit that change in transmissibility. But I suspect, it's obviously going to be an area of very intense research over the next weeks and months, and we'll get some insight into it. Whether we'll solve it completely, science is always a little bit of a temptress in that regard of, allowing you to explore and discover certain things, but always keeping a mystery about it.
That's what makes it interesting, right?
It certainly does. Don't get too confident when you know what's going on, because biochemistry and biology will prove you wrong very soon.
Well, one of the reasons why this particular mutation, I think, caught on in terms of something to focus on is because the same mutation pops up in these two independent variants of the virus. So, based on all of the other mutations that you see in the sequence, we know that, they don't share, other mutations, but this particular one does.
Right.
And that's, I'm an evolutionary biologist, so we know when something happens over and over again, there's probably something that favors the survival of that version.
It's like environmental evolutionary selection at a molecular level, right?
Right, exactly. Mm-hmm. And so one hypothesis that I've read about is that over time, because this virus can live for a long time within somebody who has a compromised immune system, that gives that virus time to continue to replicate and collect some of these additional mutations. So I think that speaks to the need for effective treatments, because the more times a virus can replicate, the more times it can accumulate mutations.
Right, and I think that opens the door is a very heavy focus has been on the vaccines, right? Because that's the preventative intervention, right? But, I think most of the epidemiologists are predicting this pandemic is going to become an endemic. And so it's not going to go away completely. So we are going to have, thousands or hundreds of thousands of individuals continuing to contract this virus, even after we have quote unquote herd immunity because of the vaccines. And so, an additional area that will continue to be very important is palliative measures of interventions about what we can do once somebody has the virus to manage the symptomology, to keep people off of ventilators, from keeping them from progressing to the severe, lung stress. So, in addition to the vaccines, there's lots of companies also looking at that symptomatic and palliative care for those individuals. And to your point, it not only reduces the the risk to that individual patient. But the more that we can control that, the less residence time that virus has in the body. And to your point, the less time to continue to mutate and adapt and try and figure out ways around our treatments and become resistant to those palliative care options.
Right. And I just want to follow up on that to clarify a point that we have from the audience that vaccines are not treatments. And that's right. Like vaccines preventative, whereas we're continuously learning about different techniques to actually treat the disease once you have been infected.
Yeah, so a very good call out. Yeah.
We have another question just about the vaccines in terms of whether they've been tested on people over 75.
Yeah, I don't have the exact details. I mean, obviously the Moderna and so it's the Moderna and the Pfizer BioNTech are the two mRNAs. And then you've got the AstraZeneca Oxford and now the J&J adenovirus, also RNA, but double-stranded RNA viruses. My understanding of those trials is that they were reasonably well designed to include a good stratification of patients, whether that be due to age distribution, ethnic distribution, or otherwise. I think areas yet to be explored, right, are individuals that are pregnant, right, to understand fetal maternal risk. And then moving into the younger populations, the pediatric populations, are things that these companies are committing to do now that they've got what I'll call the foundational body of evidence that these things work. So even though these trials were quite large relative to the amount of time that they were executed in, which is a kind of indice of the pandemic in and of itself. But over time, these companies will continue to expand their well-controlled trials, continuing to diversify the populations that they're exploring and get that body of evidence.
Right, and I've looked at the data from the data from Moderna and Pfizer that they submitted to the FDA to get that approval for that emergency use, and that's publicly available, so you can see the demographics of all of the people who were involved in the trial. And I think they paid particular attention to enrolling older people because we know that older people are more at risk.
Right, and I think also underrepresented minorities was a key focus.
Right.
Recognizing that in the past that they may have been underserved with previous vaccines. So a quite purposeful study designed by all the companies, at least to date.
Right. So, we talked a little bit about the effectiveness of the vaccine. So right now, what we know, as you addressed, is that there are some early studies looking again at that particular mutation, showing that I think the Pfizer data looks pretty good in terms of their vaccine continuing to be effective. But that's something that will continue to change. So what do vaccine companies do to kind of in this race to stay one step ahead?
Well, it's a game of whack-a-mole, right? Well, I think there's a couple of things, right? First is having a diversity of vaccines rather than just having one. No one vaccine is going to be able to treat all patients, right? Especially when the virus, has the ability to mutate. So one of the things that will work in our favor is the fact that, we now have 4 at the forefront of availability to approve, 2 on the doorstep of approvability. I think I've lost track of the number, something like 130 at one point in time that were being evaluated. Obviously that will decrease over time because not all of them will be as effective as we hope. But I think, the end state is to have a cross section of different vaccines, recognizing that as we do get additional mutations, some may be avoid some of the vaccines, but not the others. And so having the ability to not have just one option, but a series of options will be very important. And then the other piece, as you highlighted, what was done initially in the UK and now is spreading around the world, is now that we've seen these mutational effects, the scientific rigor that will be brought to continuing to monitor the evolution of the virus, will be increased to try and stay at least roughly in pace with those mutations and whatever attributes they bring to the virus itself. You can't predict them a priori, so you're always going to be in a reactive position, but keeping on top of that vigilance will be key to the future, I think.
And I think to your point, just to add, one more step of detail is that even though we have so many vaccines in development, a lot of them still focus on that one spike protein, which is, in the picture that's everywhere. It's like that little red, that red protein that's sticking out of the gray ball. Yep. And so, one of the focus of future development will be to look for other targets, because, if the spike protein changes, then, if all of our eggs are in one basket of the spike protein, then we'll need to figure out how to find other targets.
Right. And I think that will evolve over time. And I think, because this is of the SARS, virus classification, the anticipation is, we're going to have this, forever in perpetuity. And that will continue to evolve. And again, that diversification of the armamentarium of vaccines will be very important.
All right, we've got a couple of questions from the audience that I want to pause and address. First, will a vaccine stop you from being able to transmit the virus to other people?
I think that's an area of active study.
Right, this is a $1 million question right now.
I don't know, I don't recall seeing any, what I would say, conclusive body of work that's answered that question yet. Right. But it is one that people are paying, very close attention to, along with other questions like, if you get COVID once, can you get it again? These are things that only time and, part of it's time, but part of it is also getting enough data that you're confident in your conclusions from a statistical standpoint.
Great. And then the second question that we have here is, do you see a difference between the vaccines that have been approved and potential long-term side effects that people might have concerns with?
Well, I think only the data will tell you that, right? But I think what we can have as reassurances, by and large, over time, across a diversity of vaccine types, vaccines have largely been very safe and effective. So we have a huge body of, when we administer a vaccine and eliciting an immunological response in humans, that by and large, except for individuals that are hypersensitive, these are relatively safe treatments, not therapies. And the other part is that I think the other part that has not been as prominent in the public literature is that while both the Moderna and the Pfizer vaccines are the first mRNA vaccines to have been approved for regulatory use, there has been a body of work that spans at least 10 to 15 years of other mRNA vaccines, right, for things like Zika and cytomegalovirus. I think rabies was one of them. I think even some indications in the cardiovascular area. So there's a larger body of work behind the COVID vaccines with mRNAs that that suggest that long-term effects are a relatively modest risk. It's never zero, right? We won't know until we have the data, but on a risk-benefit analysis, they are heavily weighted to the safe and effective end of the curve.
Right. And I think what we've seen so far, we've seen, hundreds of thousands of people have gotten the vaccine already, with a very, very small number who have had an allergic response.
As a little fun fact, my mother, an octogenarian, received her first dose of the Moderna vaccine yesterday.
Excellent.
And she's doing fine.
That's great to hear. But I think, again, as you said, the risk is non-zero. And so, some of the precautions that are in place are that, when you get the vaccine, you're monitored for, I think, half an hour because the most common side effect that happens is that, anaphylaxis, that allergic response that really happens very immediately.
Yeah.
And so people are ready with that, the EpiPen or that treatment to tackle that right away.
Right. And when we see the public, news on this, they tend to focus on those isolated cases where there is a very significant. But when you look at them on the hundreds of thousands now of people across the globe, with the UK and the US and some other countries, is the rate of those severe reactions or even the modest reactions is extreme. It's a fraction point, and people know what to monitor for, and so the immediate risk is actually very, very low.
Right. And so, kind of an indirect question to that has come up before is that, how do some of these mRNA vaccines, because they're, newish technologies, do they interact with your DNA at all?
No, right? So the mRNAs, right? They're encapsulated in lipid nanoparticle that allows it, once injected, to get past your immune system and into the cell. It doesn't enter into the nucleus. The lipid nanoparticle kind of falls away, and the mRNA is taken up by the ribosome, which translates it into in this case, the spike protein, right? Which then elicits. So it never gets into the nuclear compartment, and so the risk is extremely low there.
Right. So your own DNA lives within a subcompartment of your cell. That's the nucleus. This falls apart before it ever, it would never interact.
Right.
With
and that's that's the characterization that's gone on in the last 20 years with the other mRNAs to understand, not only the effect of the mRNA to elicit the desired response, but also characterize any unanticipated adverse effects, as well as understand what your body does to that lipid nanoparticle and the mRNA once it gets inside.
Right. So let's switch gears a little bit, because the other story that's really been in the news is thinking about the production and the distribution of vaccines. And this gets at, at your area of expertise. And before we kind of talk about that process, I wanted to ask you to tell us a little bit about yourself, because some of your early work actually was in developing drugs for diabetes, and you've told a story before about how that really had a personal effect on you. Can you share that story with us?
Sure. So I'm the first of my family to go to college, so I didn't get a lot of guidance from my immigrant parents. And so my first exposure to kind of college education was friends of the family who happened to be pharmacists at the local university, pharmacy students. So their impact on me is, as I was going to go be a pharmacist like them. I got into college. That wasn't the right fit. I became a chemist, biochemist. And then I eventually became a mechanistic enzymologist, and then joined the pharmaceutical industries after my PhD. So in the late 90s, I was in a drug metabolism organization for Pharmacia and Upjohn at the time in Southwest Michigan. And we acquired an anti-diabetes drug called pioglitazone. And so at that time, I was a group leader, and we were responsible for developing the drug metabolism registration package for that molecule, which after we had done all of that and registered it, we decided it didn't fit our business model. So we sold that to Eli Lilly in Indianapolis, Indiana. And the compound became Actos, was the brand name. Well, my sister works for Eli Lilly, and she was on the product launch team for Actos. And coincident about that time, my father was diagnosed as a adult onset type 2 diabetic, and guess what drug he went on? That was very effective for him. So, when I joined the industry, I was motivated by the challenge and the interest of science and scientific inquiry. As I matured in the first, third of my career, it was understanding that science could benefit science and patients. And those patients were anonymous, right?
Yeah.
And the association of both my sister and myself with pioglitazone and the impact of my father, that patient is now, family and friends. And so, that's a really great motivation to come into work each day.
I love that story because, we see all these really long chemical compounds and, all of these, images of labs and, decades of research. But at the heart of it, science is all still just people, and we're doing it for the people that matter to us.
Yeah.
And I love that story that helps us understand how people get into science and what motivates you. So thank you for sharing that.
It was an amazing life blessing.
And so some of your work has, and I think you started to talk about this a little bit before, is this idea of what happens to a drug once it's in your body. So not looking at what's happening to your body, but what's actually happening to the drug. And maybe this is something that you were talking about that with vaccines is not always known.
Right, yeah. So, I've been a drug metabolism scientist most of my career. That is focused, your body is built to recognize when things foreign to it, get inside your bloodstream and your body, that it has defensive built against that. And in defending against those, what we call them xenobiotics, those foreign entities that enter your body, those systems will look to modulate those chemical entities to get rid of them and rid the threat to the body, even if they're intended to do good, right? So this was learned, mostly through the small molecule therapeutic space, right? Where when you take a drug, it's got to get through your GI tract. The first thing it hits is your liver. Your liver is your first line of defense. It's full of enzymes that look to take those molecules and make them more water soluble so you can pee or poop them out, right? Bottom line. But in so doing, it might make them more efficacious. It might make them toxic. And it may mean that they interact with other drugs that you're on, since, many of us now are what we're called in a polypharmacy state. We're on more than one medicament. Right. And so they interact with each other. And so if I give one drug, my other drug concentrations may go up, and, I might get more toxicity or less efficacy if the concentrations go down. Now we're taking those principles that we learned in the 70s and the 80s and even the early 90s on small drug to biologic drugs and now vaccines, and trying to understand once these therapeutics, whether they're preventative or palliative, once they get inside the host mammalian system, how your body manages those, right? And vaccines are actually, elicited to your immunological response is exactly one of those defense mechanisms to clear your body of these invaders, right? And that's what the vaccines are trying to do. We're trying to send a Trojan horse into your body, said, here's an imminent threat. It's actually not an imminent threat, but when the real threat comes along, you've already got, your antibodies there to defend your body.
So we're, in a way, trying to trick some of your defense mechanisms.
Exactly.
So that the drug or the vaccine can do its job.
Right.
And that's what's involved in, it's great to have, that mRNA sequence for the spike protein, but how do you package it? How do you turn it into a product that not only can, get past all of those defense systems, but can be manufactured and stored and shipped and all the way down the chain? So what's involved in figuring that out?
A lot of really fundamental science. And I think, the discussion that we were having is all that, well, I'll call it, exploratory science, even failed science on some of the other mRNA vaccines that taught us, what worked and what didn't work, put us in a position with the COVID threat that we had, some idea of how to package that, right? But it's fundamental hypothesis-driven science that is kind of below the surface of all that enables you to go from that concept to that application, right?
Right.
And and it's true for any therapeutic, whether it's an antibody, monoclonal antibody, or there's a lot of failed science behind that's taught you what will work or what's not. I think it's a Thomas Edison quote around all of his failed experiments, right? As is about, they weren't failed experiments. They taught me what won't work that led me to what will work, right? And I think that's the principle here.
Yeah, that's such a good philosophy for science. And, and I think what we've seen so far is that when we're in this race against time with this virus, sometimes you can't wait to get to that perfect state. And this is why, we're moving forward even with these vaccines that need to be shipped at ultra cold temperatures because we just need to get it out there.
Yeah. Well, I mean, the COVID situation and the mRNA vaccines are, it's a paradoxical confluence of a lot of the right factors at a moment in time to enable what you could argue is a breakthrough or a step function technology, right? So one, you had 1 to 2 decades of fundamental science behind this on other mRNA vaccines. And none of them really got the traction because there wasn't the sense of urgency. It was hard to do the clinical trials at numbers. They were focused on, rabies or Zika that are reasonably well, controlled at this point in time. So with the COVID, you have an unmet medical need that is a crisis that, has a sense of urgency, built on a foundation of, 2 decades of those experiments, some working, some not. The second piece is you've got the opportunity for some really important and powerful partnerships. So if you look at the Pfizer one, it's the partnership between BioNTech, a small, a biotech company, and an established, drug discovery development machine in Pfizer, right? And on the other one, you've got the partnership between Moderna, and in this case, it was the NIH and Dr. Fauci and the partner, enabling those investments to, do some investments at risk, given the urgency of it. And then the other part is because of the pandemic, you got the ability for kind of executional efficiency as you could enroll, dose, and evaluate, tens of thousands of patients in a fairly short period of time to get your statistical significance of what worked and what didn't. And you've got the governments coming together and kind of putting up, in a bipartisan manner, right? Is saying is that this is a societal risk, whether it's national or global, that we're going to invest at risk to share some of the burden with the companies to allow us to stockpile those unproven vaccines. So once proven, they're ready to go. And now the next phase is, as you say, is like go from our production capability of what is X today to X plus Y plus Z in the coming months.
Right. No, I think that's a really good point, because when we reflect on how quickly we've gone from, just having the genome sequence of the virus to, putting shots in people's arms, that I think, people, feel a little nervous about that. But when we break it down, that it's all of these factors that came together to really put together processes that have been in place for a long time. I think that feels a little bit more reassuring.
And I think the other thing that's helped the cycle time for both Moderna and Pfizer because of the nature of the mRNA, these are chemical processes. Right? Whereas most other vaccines require like a bioreactor for microorganisms to generate your vaccine substrate, and so that has significantly enabled the mRNAs, and is on the one hand a compelling advantage for cycle time. I think the Achilles heel is their chemical stability, fragility.
Right. So it looks like the next 2 vaccines that will be coming out for public distribution will be the AstraZeneca and the Johnson & Johnson vaccines, both of which use that delivery package of a different virus.
Right, the adenovirus, right.
Right, to bring the spike protein.
And the advantage there, right, is it looks like when you kind of tease through the AstraZeneca, they can achieve similar efficacy, which is positive. And their, supply chain, we call it the cold chain or warm chain or cool chain, depending on what your is, they will have, a much greater ability and flexibility because of that. There's still room for improvement, right? Because obviously with these cold chain, the developed and the Western world can handle these quite well, but this is a global pandemic, and we need solutions for sub-Saharan Africa and other warm, humid climates where they may not have the infrastructure for that cold chain. And that's why I think there's still much more room for additional vaccines for not only the resistance point we were talking about earlier, but supply chain considerations, as well as maybe opportunities to find vaccines that will be successful on one dose and not require a 2-dose regimen, which will increase compliance.
I have to say, I did read an interesting article that made me laugh about what vaccine distribution can learn from Dippin' Dots ice cream. In terms of being able to take super cold ice cream and put it in every amusement park.
I hadn't really thought of that one. I have to reflect on that one, but it is amusing.
So I wanted to end thinking about, where we are right now. We're facing, we've been facing over the last few weeks these issues of, logistics,
right.
And trying to figure out how to balance supply and demand. We're seeing some policy changes, rolling out this week, looking at expanding the pool of, who's eligible to get vaccines, starting to release all of the available doses right now to try and reach more people faster. So what do you see in terms of other potential solutions to the situation that we find ourselves in right now?
I don't know that I have any novel insights. I think it's going to be the collective will, wisdom, and learning kind of across the board. The one thing that I think is, unique to this period of time, the amount of sharing that's going on between, governments and companies to share best practice for the good of society bodes well for us. It's not being kept isolated. And every day, we're getting additional experience and learning, whether that's in the manufacturing sector, whether that's in logistics, or whether that's in the administration opportunities. So, I think there's an aligned collective will here. There is not going to be any one solution, but success is going to come through, hundreds and millions of mini solutions that creative individuals around the world problem solve in their specific situation. So I'm a pathological optimist, but I think there's a lot of evidence to say that it'll be painful, we'll stub our toe, it won't be as fast as we would want, but I think it will accelerate as we get more and more experience.
So a very practical question from somebody, for someone who's 64 years old, is there an advantage or disadvantage to using any of the vaccines?
Not that I'm aware of at this point in time, right? I think the disadvantages don't take it. The advantages take one of them. Everything I've seen, and but it all should be done in consultation with your physician for your own personal health situation. So always under consultation. But I think by and large, at the highest level, you're at greater risk not taking it than you are taking it. And which one, at this point in time, hard to discriminate.
Especially with the levels of the virus,
right.
as they are in our community right now.
Right.
But I think one solution that has caught my eye just as a local story, I've just read about a nonprofit organization called Philly Fighting COVID, who is working with the city to organize a mass vaccination center in the convention center.
Cool.
And we're hearing about, places like Disneyland and baseball stadiums.
Dodger Stadium, yeah.
Right. So I think you're exactly right that, the as much as you can try to plan for these kinds of things, some that you just have to learn as you go along.
We've never faced anything like this in, our contemporary life. The last time probably was the mobilization during World War II, and that didn't happen overnight, in the manufacturing sector or even in the pharmaceuticals. It was the penicillin, and sulfa drug production. So, mankind and civilization has a recurrent history of solving these things that I think, it's good not to lose sight of.
Right. And in the meantime, we just continue wearing our masks and staying distant.
Yep. Masks, distance, hand washing.
I think, that's, circling back to where we started, no matter what strain of virus we're dealing with, those things work. So let's get the vaccine out as quickly as we can. But in the meantime, we all still have to keep protecting each other.
Agreed.
Well, thank you so much for joining us today. It's really interesting to get your insight into some of those logistical aspects of starting from that, first target, genetic sequence or molecule and thinking about what it takes to build all the way up to where we are now with vaccines. And so hopefully we're at a turning point soon.
I believe we are.
So
thanks so much. It was fun.
Thanks everybody for joining us and for sending your questions, and we will see you back again next month. And I'll share that we are excited that the Franklin Institute is open again. So we are operating under very safe conditions, and so please come visit us if you're looking for something to do. Thanks again, and we'll see you soon. Stay safe.